Hello, today I will be presenting a coordination declaration using ESR to elucidate the coordination sphere of copper crystals. In particular, we will be looking at three unique copper-2 crystals that we have synthesized in the lab. So let's get started. So we will begin with a brief introduction on the biological significance of copper-2 in aerobic organisms. So copper is a central nutrient in organisms that require oxygen. Copper is able to participate in fascinating Fenton-like chemistry, which shuttles electrons to and from chemical species by switching between its cuprous and cupric states. On the left is the amino acid histidine with its R group imidazole. The lone pair on the imidazole nitrogen is capable of coordinating very strongly to transition metals, including copper as shown here. Take note of the electronic configuration of copper-2 ions for now, being D9, as a further discussion of the importance of this configuration is soon to come. So here's a more sophisticated example of a copper-2 histidine coordinated enzyme. This would be the cytochrome C oxidase, which is shown in the red square. Cytochrome C oxidase is present in the electron transport chain, which is a schematic of it is shown on the left here. It is vital to the production of ATP in aerobic organisms, which is the cell's main source of energy. So if we zoom in here, you can see through the copper ions that they are coordinated by various ligands, including imidazole, oxygen, and sulfur from the various R groups of amino acids. So using ESR, we are able to hypothesize what type of ligands are surrounding the paramagnetic metal species. So now that we understand some significance of our study and copper itself, let's dive into the theory of ESR and what it is exactly. So as you may know, electrons have the intrinsic property known as electron spin, which means they behave kind of like a bar magnet in a magnetic field. They can exist in the spin up alpha position or the spin down beta position. The alpha position is of higher energy than the beta position when exposed to a magnetic field. When a unique quantized amount of energy, which we will call h nu, is applied to the sample present in the magnetic field, the free electron, which keyword here is free electron, the species must be paramagnetic, the electron is capable of promoting to the alpha position and then back down to the beta position. This phenomenon is known as electron spin resonance, or ESR. So now knowing that unpaired electrons behave this way, we see that the cupric ion's D9 configuration is actually what makes the species ESR active, since it is paramagnetic. So we now consider dppH, which is shown on the left. The electron in dppH is delocalized throughout the entire compound's pi system, making any nuclear spin effects on the electron negligible. Because of this, we use dppH in our experiments as a model for the free-moving radical electron. The figure on the right here is the Zeeman splitting of the zero nuclear spin compound when it is exposed to a magnetic field. So when the energy difference is met here, electron spin resonance occurs and a peak is recorded by the instrument. Now, another important factor to consider is the Boltzmann distribution. Because we reside in the lower energy state first, and then the electron is promoted to a higher energy state, we must have a slight excess, at the very least, of electron density in the lower energy state. So using the equation shown on the right, which the terms here are the G factor of dppH, the Bohr magneton for an electron, and the field strength of the center field, respectively, we are able to solve for the Zeeman splitting energy and solve for the other ratio of electrons in the higher to lower state using the Boltzmann equation. And as you can see here, this value is less than 1. It's 0.998 to be exact. So this means that there is a slight excess of ground state electrons, which, as I said previously, this condition is necessary to observe the ESR phenomenon. When considering the effects of nuclear spin on ESR spectra, things become a bit more complicated. Since copper has a total nuclear spin of 3/2, it falls into this category. From quantum mechanics, we take the total energy of the system, shown here. Since there are 4 different combinations of Ms and Ml, we can obtain 4 energy solutions for the equation, shown in the left figure. Because the Bohr magneton of the proton is smaller by 4 orders of magnitude than that of the electron, we are able to cancel out those terms, which is a common practice in ESR experiments, as they have an insignificant effect on the results. Applying the solutions to the existing Zeeman splitting diagram yields a much different result, as shown in the figure on the right. We now introduce two new terms, the G-factor and the hyperfine coupling constant. The G-factor is a proportionality value that relates the magnetic moment of a compound to its spin angular momentum. A larger G value will correspond to a more diffuse d orbital, directly caused by the presence of electronegative ligands. The hyperfine coupling constant defines how tightly localized the electron is to the metal nucleus. As this value decreases, the electron is delocalized to a greater extent. Again, ligands directly affect this value, as you will see shortly. Now that we've introduced some of the theory behind ESR, we will begin with the experiment. We start with the hypotheses that were formulated prior to the experiment. The first hypothesis is that the Lane G factor for all three species, as shown below, will be greater than dppH. Further, because substance 2 has the electron withdrawing carboxylate ligands, it will expand the copper d orbital the most, and we should observe the largest G value since it will have the largest spin angular momentum as a result of the expanded d orbital. The second hypothesis states that as the number of electron-donating imidazole ligands increases, we should observe increasing hyperfine coupling constants since the lone electron will become more localized to the copper nucleus as the electron-donating imidazole ligands increases. So here are the syntheses for the three samples here. They were each synthesized using the corresponding copper to reagents on the left of the reaction arrow. Sample 1 utilized diethylene triamine in the condition shown below the reaction arrow. Sample 2 utilized 2-methyl-imidazole as well as the condition shown below. And finally, sample 3 used imidazole and simply the reaction condition shown below. Next, these samples were all dissolved in a glycerol sample solution, 25% glycerol, 75% sample, and loaded into ESR tubes, which were submerged in liquid nitrogen, loaded into the instrument, and the spectra were recorded. So here are the spectra obtained for the 4 samples taken. There are 2 important values obtained from the 3 sample spectra. So first is B center field for each sample, including dppH, and this is necessary to calculate the G factor, which is done later. Next is the hyperfine coupling constant, which was calculated by averaging out the difference between each peak. The values used are shown here as the red marks, and you can see the straight bars that are not dashed, the distance that is separated by the dashed lines, was taken and averaged out for each spectra, and that average value was the hyperfine coupling constant. So as you can see, samples 1 and 3 are actually missing a discernible 4th peak each. And this is likely a leading contributor to error in our analysis. It is possible that these peaks could potentially be resolved by increasing the signal-to-noise ratio, but we are unfortunately unable to do this due to just instrumental limitations as well as time period constraints. This figure contains all the important values that were sought and obtained experimentally. The G factor of dppH was obtained using the equation on the middle left and the set instrumental scanning frequency of 9,600 MHz. Using the center field value from dpPH's ESR spectrum, its G value was determined to be 2. The G values for the other samples were obtained using the equation on the middle right, which is a function of the product of dpPH's G value and its B center field, divided by the B center field value of the sample of interest. To determine the center field value for each sample, half alpha was added to the second peak for each spectrum. This is because, since theoretically we should have observed 4 peaks for all of the spectra, even though some only showed 3, the 4 peaks should have been observed, so we added half alpha to the second peak to get the center field value. Further, the hyperfine coupling constant for each sample was determined using the methods described on the previous slide. Interestingly, we observe support for our first hypothesis since substance 2 has the largest G value and substance 1 and 3 are both larger than dppH's. This is due to the physical expansion of the copper d orbital as a result of ligand coordination by electronegative atoms. Additionally, partial support for our second hypothesis is also reported. While alpha does increase with increasing imidazole ligands, it appears as if there are other factors which must be considered when determining alpha, such as the presence of other strongly electron-withdrawing ligands like in substance 2. The electron-withdrawing carboxylate ligands in substance 2 very likely contribute to its smaller alpha value, meaning the electron is more delocalized from the copper center. While no alpha value for DPPH is recorded, there is certainly nuclear coupling that occurs. The reason we cannot observe it, however, is because the electron moves so freely around the delocalized pi orbitals that nuclear perturbations simply do not affect the splitting enough to record a difference, which is why no alpha is observed or reported. Finally, it is interesting to note that the alpha value and G value are inversely proportional, as seen in the data. This is because as the G value increases and the d orbital expands, the lone electron in copper is less confined to the nucleus within the expanded d orbitals, and the alpha value becomes smaller. It makes sense that the two values would be inversely proportional given the nature of what each value reports. So returning to our original example of cytochrome C oxidase, you can apply ESR techniques to support or challenge the reported coordination of the copper center. Synthesizing a copper crystal coordinated to 1 imidazole, 1 hydroxyl, and 2 sulfur ligands should reveal similar values of ESR data of the enzyme's copper subunit. Sort of like an NMR experiment, but using G values and alpha values to confirm structure instead of chemical shift peaks. Additionally, we can compare the G and alpha values obtained from the experiment to reported values to learn how tightly localized the electron is to the copper center. This would reveal interesting properties of the reaction site of the enzyme's copper pool, permitting researchers to determine if it is likely that the redox chemistry occurs at the copper center or perhaps somewhere else along the coordination sphere. In conclusion, we've learned that introducing ligands to a copper-2 center can create a unique spectra for many versions of copper-2 crystals, and as electron-withdrawing ligands are added to these crystals, an expansion of the copper d orbital is noticed, as well as electron delocalization. Moreover, introduction of more histidine amino acids, which contain the imidazole ligand, introducing more of those residues around a copper center serves to confine the electron tighter within the copper's contracting d orbital. Finally, the G factor and hyperfine coupling constant can be used as identifiers of the metal center's coordination sphere and its chemical environment. Thank you for listening to the ESR PowerPoint, and I hope you learned something.